Okay, day 4. We looked at the water column yesterday. Now the goal is to look at the animals that live in that water column. So we're going to look at scientists that have been pulling out some of the animals from the depths all the way from the bottom of the ocean all the way actually to the top of the surface. So let's go look at some of those scientists now.
Here in the Santa Barbara Basin, we have the bottom of the basin is very low oxygen, as really has been the main mission to explore that. And there are very few animals there. But as we go up the slope, in the more oxygenated water, we find much more diversity. And today we've hit some of the higher oxygen waters with a box core, and that's what I'm doing right now, is sorting out all of the different animals from there. And then we photograph them, and we preserve them in various ways for study. And ultimately, those animals that we've collected, plus the ones from the deeper cores, where there's low oxygen, are going to be used to form a baseline of data. And that gives us a snapshot of what's living today here, and we will sequence a lot of the DNA of those animals. And then we have this DNA baseline that can be then used if they're looking for old DNA in the sediments. They can check and see, well, does this DNA match what we've got today, or is it different DNA? And eventually we'll be able to build up a model and a pattern of DNA through time related to today's DNA. So you start off with really a big box core full of mud. So you've got several cubic feet of mud. And most of the animals live in the surface. They're in the top few centimeters, because once you get down 15 centimeters, 6 inches or so, there's not enough oxygen down there and most of the animals die. So we take the surface layer and then we run that through a sieve, and then we end up with this kind of mixture of organic material and animals. And then we sit here under the microscope and we use a little forceps and a pipette, and I just look through there and suck out every animal I can find. And then I end up with them in a dish here, and then I go through this dish and pick out and put them into different species. And then I really see my job as documenting that diversity in the deep sea. And so we're continually finding new species, as I said, outside of this channel where it's been fairly well studied. Wherever we go, we find new species. And so I see my prime goal as a researcher, my prime job at Scripps is to find and describe new species in the deep sea. Even in Scripps is in La Jolla, in part of San Diego in California. We even find new species just outside of Scripps, right near shore. So really, wherever you go, there are still new species to be found, and that's important. We don't know really, until we know what's there, we really can't understand how these ecosystems work and then how the whole of the ocean systems work. So that's what I see as my prime goal. And then my other job is to relate these organisms together. And so I make evolutionary trees. And that's a big part of my research, is taking all of these different animals, sequencing their DNA or looking at their anatomy, and then making evolutionary trees, naming the new animals when necessary, and then answering questions about evolution and the evolutionary process. The main thing that we've gotten most of our work from this trip has been the box core. And so that core goes down, it comes up, and it's generally preserved the surface layers of the sediments very well. And we've then been just taking those surface levels out. The multi-core was something that we were hoping would work more successfully, but the way the nature of the sediment here has meant it hasn't worked that well. What's good about a multi-core is it lets us take individual cores, and then we know exactly the surface area of the core, and we can sort out and get quantitative measures, and we did want to do some of that. But with the box core, we've been able to go in and just do cores anyway, and that's worked out just fine. So really, nearly everything I've done, all my time spent at the microscope here, has been based on the box core results.
There are fish in the ocean that inhabit a region called the mesopelagic, or the middle layer of the ocean. So if we took a column out of the ocean and we said, well, the surface is at the top, well, the mesopelagic area would be 200 meters below the surface down to 1,000 meters. Now we're in the Santa Barbara Basin, so we don't go down to 1,000 meters, and the basin ends, the deepest part is close to 600 meters, so I'm trying to sample between 200 meters and 500 meters. Now, to determine which ones are vertically migrating and which ones aren't, I'm going to tow a specific type of net at the surface, and then I will collect those samples and see what's in them, and then I will do the same net tow at a deeper depth. And obviously the ones that I get at the surface at night are vertically migrating. The ones that are at the deeper depths and not found at the surface are not vertically migrating. And we say transforming juvenile because that is the stage at which the fish transforms out of the larval stage into the juvenile stage, where it's going to start exhibiting the behaviors it will continue to exhibit as an adult. So again, I'm interested to see where the transforming juveniles exist in the Santa Barbara Basin because it's a low oxygen area, and I have a feeling that the transformations will happen at different depths relative to other areas that are more oxygenated. Now, I can't use the IKMT to collect transforming juveniles and determine what layer that they came from in the water column, because that net, when it's fishing, is open all the time. The mouth of the net is open. Now, the type of net I'll be using to collect the juveniles, or the larvae and transforming juveniles, is a system that opens and closes. So we have a system of nets, and it's a series of 9 nets that open and close. And the way that we do that, it's really fascinating. It's quite a large net, and a series of nets. And we sit here on the ship in the lab once it's been deployed, and we have a conductive cable over the side of the boat connected to the net that talks from the computer, and we tell the computer to tell the net when to close the net, so that I can say, oh, well, I know I'm at this depth, I'm at 500 meters. I'm going to open one and then close it at 400 meters. So now I know that whatever's collected in that net came from the layer of 500 to 400 meters. So if I do this a number of times in succession, then I can figure out where my animals came from. And if I can find the transforming juveniles, then I'll be really excited. And I'll compare it to other ones that I found in other areas and see if there's a difference based on the low oxygen compared to the oxygenated areas. So this jar here is from one of the Midwater trawls that I did, that IKMT net that I spoke of before. This is from one of my deep tows, so like I said, I'm sending that net down to almost the bottom, without touching the bottom, of course, and collecting animals down in the depths and comparing it to what I find or don't find at the surface layer.
Well, I'm Lindsay Sala, and I am the Assistant Museum Scientist and Collection Manager for the Pelagic Invertebrates Collection at Scripps Institution of Oceanography. I'm here for a couple different reasons. One is there's several graduate students that are collecting samples, so I'm here learning some of the methodology that they're using and also helping them with their samples. So basically they have several different research projects going on, and then once they're done with those projects, those samples get accessioned into my collection, in which I require the samples and the data, and then we put this on an electronic database that becomes available worldwide. So even after their projects are completed, then people, if they're interested in this geographic location and how we collected these samples, they can then use these samples for other projects. So the zooplankton collection has over 126,000 samples currently, and we're trying to continue to maintain posterity for future use and future projects. So I have a couple different things here that we collected. These jars, these first 3, we actually collected on a more surface tow with an IKMT at about 20 meters. A lot of these animals are more gelatinous in nature. Here you have self chain, which all these individuals connected into a colony. These are actually in phylum Chordata, which is the exact same phylum as humans, so this is one of your closest relatives in the sea, with a notochord and a dorsal hollow ventral nerve cord, some derived characters. These guys are filter feeders, so they're feeding on phytoplankton. They also have extremely fast reproduction, so they're a really good indicator of how the ecosystem's doing, and if there's a lot of phytoplankton in the area, these guys just completely take off. Also have a hula skirt siphonophore. This is within the phylum Cnidaria, so a relative of jellyfish. When we first collected it, it was bright pink and orange. It was a really beautiful animal. Unfortunately, once it's preserved, it sort of takes out some of the color. Okay, and this is a pyrosome, also collected on the surface. This is a colonial animal, but also in the phylum Chordata, so same as us, close relative. These are filter feeders. We actually have cited one that was 15 feet in Mission Bay. Also, there have been some cited up to 30 feet in southeastern Australia, so these animals can get extremely large. But not to worry, they're just filter feeding, so nothing that we need to be concerned about. And then here we have a sample from a Mach Ness. This is an assortment of zooplankton, a lot of base of the food chain animals in here. We have copepods, euphausids, some other types of shrimp, doliolids, siphonophores, so a whole variety of animals in here. Phytoplankton as well, which a lot of these animals are eating. So we did actually find a small octopus. That was quite interesting, but coming to the surface probably to feed at night. So we kept that till the morning to show everybody, and everybody's kind of excited and got to hold it and take some photos with it. So it's nice to interact with something like that.